my friend. Hello, guys. Да, мы сегодня вот так, да, вот так. Я today I study English. I study English. So, oh, okay. All right. Guys. Okay. Continue. There. Here. Okay. Here. Here stays. Stayed. Here stayed. We stayed here. So, alright. Okay. Oh, ito. Okay. Oh, siya. Okay. Start. Overview of acharotic cellular it's die. It's die. Me. This is word. Cellular. Okay. Cellular organization and function. Functions. Huh. I don't know it. I wasn't know it. Okay. Functions. Overview of acharotic cellular organization and functions. This section previews the major constituents and processes. Okay. Up. Processes. Okay. Overview of acharotic cellular organization and functions. This section previews the major constituents and processes of acharotic cells. With this background, the reader will be able to appreciate how
Appreciate.
appreciate,
Appreciate.
appreciate,
Appreciate.
appreciate. Um. Okay. Processes. Function processes. Appreciate. Okay. Appreciate. It's cool. Plasma membrane. The plasma membrane is the interface of the cell with its environment. Oi.
Owing. Owing.
Owen.
Owing.
Owen. Okay. To the hydrophobic interior or of its lipid barrier, but this is terrible.
Bilayer.
Bilayer. Oi. Hi, mama. Bilayer. Bilayer. The plasma membrane is impermeable to ions and most water soluble.
Soluble.
Soluble. Water soluble molecules. Consequently, they cross the membrane only through transmembrane channels, carriers, and pumps. These transmembrane proteins provide the cell with nutrients. Okay.
Nutrients.
These transmembrane proteins provide the cell with nutrients, control internal ion concentrations, and establish. Oh my god. Establish a transmembrane electrical potential. A single amino acid change in one plasma membrane pump and okay. Cl minus. Okay. chlorine. Okay. Chlorine. Chlorine. Okay. Channel causes the human disease cystic fibrosis. Okay. Let's find.
Human disease cystic fibrosis.
Fibrosis.
Human disease cystic fibrosis. Human disease cystic fibrosis.
Human disease cystic fibrosis. Channel causes the human disease cystic fibrosis. Other plasma membrane proteins mediate interactions of cells with their immediate environment. Transmembrane receptors prevent the binding of extracellular signaling molecules such as hormones. Oh my god. Okay.
Hormones in growth. Hormones in growth.
Growth. Hormones.
Hormones in growth. Hormones in growth.
Hormones and growth. Hormones and growth factors interchemical or electrical signals that influence the activity of the cell. Genetic defects in signaling proteins which mistakenly turn on signals for growth in the absence of appropriate extracellular stimuli contribute to human cancers. Plasma membrane adhesion. Oh my. Oh my god. Adhesion proteins allow cells to bind specifically to each other or to the extracellular matrix. These selective interactions allow cells to form multicellular associations. His full associations. Wow. Multicellular associations such as wow. It is cool. Such as epithelial, yes. Okay.
Epithelia.
Epithelia sheets of cells that separate the interior of the body from the outside world. Similar interactions allow white blood cells to bind bacteria so that they can be ingested and killed. To bind? Bind. Okay, alright. Alright. Ingested. Ingested. Associations. Bind are ingested and killed in cells that are subjected to mechanical processes such as muscle and epithelial, cytoskeletal. How reading it? How is it reading? How's it reading? Cytoskeletal. Filaments. Filaments. Okay. Filaments inside the cell reinforce the plasma membrane adhesion. Proteins in skin defects uses some attachments instead. Okay. What? Attachments cause blistering disease. Natri, kali, glucose. Wow. Glucose. Nasal cytoplasm. Actin. ATP. Natri, kali. Okay. Figure. I don't. I don't remember. I don't remember. Okay. Figure structure and functions of an animal cell plasma membrane. The lipid bilayer
Bilayer.
Bilayer is a permeability barrier between the cytoplasm and the extracellular environment. Transmembrane adhesion proteins enter. Let's find. Anchor the membrane to the extracellular matrix or to light receptors on other cells, and transmit process to the cytoskeleton. Transmit process. B. Oof. Adenosine triphosphate ATP-driven enzymes pump concentration gradients to transport of nutrients into the cell. Selective ion channels regulate the electrical potential across the membrane. Large variety of receptors bind specific extracellular ligands and send signals across the membrane to the cytoplasm, nuclear envelope, nuclear pore, nucleus. I know it. Nucleus, and nucleolus. Chromatin, electron micrograph of a thin section of a nucleus. Harvard. It's cool. Nucleus. The nuclear envelope is a double membrane that separates the nucleus from the cytoplasm or traffic into and out of the nucleus spaces through nuclear pores that bridge the double membranes. Inbound traffic includes all nuclear proteins and ribosomal proteins destined. I don't know. Destined for the nucleus. Outbound traffic includes mRNAs and ribosomal subunits. The nucleus stores genetic information in extraordinarily long DNA molecules called chromosomes. Remarkably, portions of genes encoding proteins and structural RNAs make up only a small fraction of the 3 billion nucleotide pairs in human DNA, but more than 50% of the 97 million nucleated pairs in a nematode worm. Regions regions of DNA called telomeres stabilize the ends of chromosomes, and other DNA sequences organize centromeres that direct the distribution of chromosomes to daughter cells when cells divide. It's cruel. Divide much of the DNA encodes alright. Myriad. Okay. Myriad of RNAs with the regulatory activities, the DNA and its associated proteins are called chromatin interactions with histones and other proteins fold each chromosome compactly enough to fit into discrete territories inside the nucleus during mitosis. Mitosis. Okay. Mitosis. Oops. Mitosis. Chromosomes condense and reorganize into separate structural units suitable for sorting into daughter cells. Regulatory proteins called transcription factors tune specific genes on and off in response to genetic, developmental and environmental signals. Enzymes called polymerases make RNA copies of active genes in a process called the transcription. mRNAs specify the amino acid sequences of proteins. Other RNAs have structural, regulatory, or catalytic functions. Most newly synthesized RNAs are processed extensively before they are ready for use. Processing involves removal of intervening sequences, alteration of bases, or addition of specific chemical groups at both the ends. For cytoplasmic RNAs, this processing occurs before RNA molecules are exported from the nucleus for nuclear pores. The nucleus assembles ribosomes from more than 50 different proteins and free RNA molecules. Genetic errors resulting in altered RNA and protein products cause or predispose individuals to many inherited human disease, ribosomes, and protein synthesis. Ribosomes catalyze the synthesis of proteins using the nucleotide sequences of mRNA molecules to specify I feel good. Do 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 do. Do 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 do. Okay, continue. No control. Da 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 da. Okay. Ribosome and protein synthesis. Ribosome catalyze the synthesis of proteins using the nucleotide sequences of mRNA molecules to specify the sequence of amino acids. Ribosomes free to in the cytoplasm synthesize proteins. Okay, let's find check. Synthesize. I'm right. Synthesize proteins that are released for routine to various intracellular destinations, endoplasmic reticulum. Ribosomes synthesizing proteins destined for insertion into cellular membranes or for export from the cell associated with the ER, a continuous system of flattened membrane sacs and tubulous proteins produced on these ribosomes carry signal sequences of amino acids that target their ribosomes to receptors on the ER. These regions of the ER are called raw ER owing to the attached ribosomes as a polypeptide chain grows. Its sequence determines whether the protein folds up in the lipid bilayer or translocates across the membrane into the lumen of the ER. Enzymes add sugar. Raw endoplasmic reticulum, mitochondria, free ribosomes. Mitochondria, raw endoplasmic reticulum, a smooth endoplasmic reticulum, Golgi apparatus, lysosome. Electron micrograph of a thin section of a liver showing organelles. Harvard, Boston. Polymers to some proteins exposed in the lumen. Some proteins are retained. Retained in the ER, but must move on to other parts of the cell. ER is very dynamic. Motor proteins move along microtubules to pull the ER membranes into a branching network spread throughout the cytoplasm. Continuous bidirectional
Bidirectional
bidirectional traffic moves small vesicles between the ER and the Golgi apparatus. These vesicles carry soluble proteins in the lumens, in addition to transporting membrane lipids and proteins. The proteins on the cytoplasmic surface of the membranes catalyze each membrane bonding and fusion. Even the use of specialized proteins for bonding and fusion of membranes at different sites in the cell organizes this membrane traffic and prevents the membrane components from getting mixed up. The ER also serves as the outer membrane of the nuclear envelope, which can have attached ribosomes. ER enzymes synthesize many cellular lipids and metabolize drugs. Metabolize drugs while ER pumps and channels regulate the cytoplasmic concentration. Golgi apparatus. The Golgi apparatus processes the sugar side chains. Chains on transmembrane and secreted proteins. Each consists of a stack of flattened membrane-bound sacs. Socks with the many associated vesicles. The Golgi apparatus is characteristically located in the middle of the cell near the nucleus and the centrosome. Proteins to be processed come in vesicles that detach from the ER and fuse with the Golgi apparatus membrane so as proteins pass through the stacked Golgi membranes from on one side to the other. Enzymes in specific stacks modify the sugar side chains of secretory and membrane proteins. On the downstream side of the Golgi apparatus, processed proteins that are segregate segregate into different vesicles destined for lysosomes or the plasma membrane. Many components of the plasma membrane, including receptors for extracellular molecules, recycle from the plasma membrane to endosomes and back to the cell surface many times before they are degraded. Defects in this process can cause arteriosclerosis. Lysosomes and impermeable membranes secrete degradative enzymes inside the lysosome from other cellular components after synthesis by ER. Lysosomal proteins move through the Golgi apparatus where enzymes add the modified sugar phosphorylated mannose recycler transport guided by phosphomannose receptors delivers lysosomal proteins to the lumen of lysosomes. Cells ingest microorganisms and other materials in membrane vesicles derived from the plasma membrane. The contents of these endosomes and phagosomes phagosomes are delivered to lysosomes for degradation by lysosomal enzymes. Deficiencies of lysosomal enzymes cause many severe congenital diseases where substrates congenital Да, я вам. Нет, физика там где-то.
Нет, пожалуйста, мусор убери и...
Я мусор уже унесла туда, он уже внизу. Чашки я принесу, мам. Substrates of the enzyme accumulate in quantities and that can impair their function of the brain, liver, or other organs. Mitochondrial tomorrow. Mitochondria will tomorrow. Uh-huh. Acting. Acting.
Acting.
Acting. Acting. Ah. Okay. 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 Um. Guys. Bye bye. Bye bye, guys. Tomorrow, meet.